Hello, my name is Alexia. The Hendra virus is an emerging paroxysmal virus which was first discovered in Brisbane in 1994 after an outbreak of the disease affected 21 horses. The horses developed high fever and respiratory signs and had a high fatality rate. Two people who had close contacts developed respiratory disease and one person died. Infected cells share also structural characteristics such as the formation and presence of nucleocapsids in the cytoplasm and enclosed bodies. They form membrane-like structures in the cytoplasm and long tubules that might have a role in the RNA and have a non-segmented genome. Hendra encodes two membrane glycoproteins and an attached glycoprotein which serves to bind the neurons, smooth muscle and endothelial cell lining, small arteries, and a fusion glycoprotein which mediates fusion between virus and host cell membrane. Hendra virus phosphoprotein gene encodes several products including the protein itself and are all competitors against the type 1 response to infection. Little is known about the transmission of Hendra, however flying foxes are the natural reservoir for the virus, and experimental infection doesn't recognise the clinical disease, but the virus can be detected in blood, urine and saliva. The exact mode of transmission from flying fox to horse is still unknown, but is most likely through pasture or contaminated feed. Transmission of infected horses comes from direct contact with horses through bodily fluids. A total of 67 equine cases have been recorded near coastal regions of Queensland and northern New South Wales and one event in Chinchilla. In people, most cases present as influenza-like illnesses and then progress to a more severe illness to a relapse. Relapses can occur up to 13 months after infection, which suggests that the virus can be reactivated after inactivity or can persist after initial infection. This can be seen on the following table. Clinical features are non-specific. It is a level 4 biosafety. Hendra can replicate in various cell lines and most cases are fatal. Quantitative real-time PCR to detect RNA is currently a diagnostic method of choice due to speed and accuracy. However, in labs, ELISA tests are mostly used for possible serum samples. Now, approved antiviral drugs are available in the period of this article being written. However, in 2012, there has been a vaccine invented for horses which comes in a dosage of 3. Evidence doesn't support airborne exposure, so the virus is transmitted through bodily fluids or contact with infected materials. Such efforts have been made to educate the equine industry about Hendra and to be able to recognise symptoms and signs of Hendra. Quarantine of any infected horses or areas are done by authorities. Development of the equine and human vaccine are both important in the control of Hendra virus infection and are both potential flying fox's genetic diseases. This article is written by Zuresh Hamanish and al.